Right, so let's speak about what you've done in experiment 1 and what you need to do for your assessment. So in this, for the assessment for experiment 1, you will be writing certain sections of the report. So as you did in CMY 202, we once again slowly build up your report writing skills, and then in your final experiment, you'll be writing a full report. So for experiment 1, you'll need to make a title, have a method section, have a results and discussion section, and have references, each allocated a certain amount of marks, and then that totals 50 marks. You also will have certain assignment questions, and these are detailed how many marks they exactly count in your practical guide, and you just need to ensure that you write enough there to actually earn this amount of marks. So in other words, if the equation counts 16 marks, do not just write 2 equations as your answer, for example. So then it indicates that you really need to have a significant amount of things written there. The same with this. So you can of course see that, for example, the title and the references are just easy marks to earn, but they still need to be descriptive. The method section is 10 marks, so you just need to tick the boxes essentially of what needs to be there. And then the main focus is the results and discussion section. Looking at what needs to be in your report writing, so we start with the title. So in your title, the whole idea behind the title is always that you shouldn't copy from what the title is in the Prac manual or the practical guide. So what we give you is descriptive chemistry, right? It says experiment 1, descriptive chemistry. So your title should not say just descriptive chemistry. It should say the investigation of how oxidation state and descriptive chemistry can be combined to study whatever, or the study of something. You come up with some sort of creative title, which is around about 15 words. It's not a strict limitation, meaning it can be 16 words or whatever, but around about 15 words. If you can keep it to less, nice unique title. You can use the general guidelines at the back of your practical guide to give you an idea of how to write a nice title. And then depending on how unique it is, and if it's at least descriptive and there's no grammatical errors, you can earn up to 2 marks for that. Moving on to the method section. This would generally be around about a page. And remember, the method section needs to be like a story. So it's not written in bullet point form, it's written in paragraph form. So whoever is following along will read your method section and what you've done. So you can split this up into each experiment's method for that specific experiment. So in other words, experiment 1.1, you have a little method section. Experiment 1.2, a little paragraph on how to do it, 1.3, and so on and so on. All right. For each one of them, written in nice paragraph form. But then you must remember you're dealing with a chemist. So in other words, you don't have to say you need to add the green substance to the purple substance. You can say you're adding manganese chloride, or you're adding this acid or whatever. So don't oversimplify anything or give too much detail. You can really write as if you're speaking to someone else on a second year level and how they need to follow along. All right. Any qualitative sketches that you would like to add? So if you would like to draw something to make it nice, like I say, it doesn't have to be a work of art, but it always aids in terms of experimental setup. So if something is not really that clear in your writing and you feel a figure would aid you in this, that is a good idea. This is maybe not be too applicable for experiment 1, but especially later on, if you move on to third year or write other practical reports, it's always good to remember that you can put in a figure of the experimental setup or whatnot. All right. Then important things to note is anything that's easily misunderstood, steps that were not that clear for you in the prac, that must be well explained in this. Right. And you must always add any relevant theory, meaning of the specific methods. In other words, it's not theory meaning the theory of the practical itself, meaning, for example, you work with a redox reaction. So the redox theory will go into something like your introduction. This theory means, like, let's say you're working with a distillation setup. What is the theory of distillation that you need to use in the methods. And then you would add this theory here in the methodology section. Not really too applicable, but once again, that's just a general note on the method section. You'll also note here some general things, meaning observations or masses that you use. So for example, if you add 1 gram and whatnot. So that's essentially in your story. So somebody needs to be able to repeat what you did. So if you don't list the masses or say that you add 1 gram to 20 milliliter of this, then someone's not going to be able to repeat it if you give them some vague idea of saying add the sodium chloride to the water, because how much sodium chloride are you supposed to add to what water in what kind of glassware? So that's what you need to describe in your method section. Now, in CMY 282, we generally split the results and discussion section. And in general, you will split the results and discussion section into 2 separate sections. So you'll have a results section and a discussion section. But for experiment 1, because you have so many separate little experiments and whatnot, we've decided to group them together. So in other words, what you have is you have your results. So your results in this case will be typical observations and saying, we've added this and this, and we saw this color change, or we saw this evolution of gas or whatnot. And then you discuss, why do you see this color change? In other words, manganese in the 2+ state versus manganese in the 7+ state needs to correlate with this color change, and that is why we see that, for example. Right. So the result is the color observation, and the discussion is the chemical interpretation of what you saw. So basically, so that's why we combine it in this case, because otherwise your results section is going to look really sad, and your discussion section is also going to look really sad. But typically you would separate the two, because in a typical sense, you would have in your results section, you would have just listing of your yield, your masses, instrumental data, observations, color, physical properties in production, other things like calculated values, subsequent discussions, structures, chemical reactions, mechanisms. And then in your discussion section, you would then discuss these things that you've just listed in the results section. But now, because we combine them, you need to list those things and then discuss. Give them chemical importance. In other words, why does the chemistry indicate to you what you've done? So look at your experiment, look at what the theory says, and how does what you've observed then link to chemical theory. Okay. You need to keep this section quite concise, but please try to incorporate all the relevant information. In other words, try to use tables, and I'll speak now about tables and figures and schemes, equations, so that you summarize the information best as possible. So all the kinds of information, your results, as well as possible, and then you have a short paragraph on each sub-experiment. So meaning experiment 1.1, 1.2, 1. Whatever, so that you have a short discussion. So I would typically have a results and discussion, main heading, and I would say experiment 1.1 results and, or I should say results and discussion, experiment 1.1. I have my little table of the summary of the things that I saw, and then I would have a discussion of what would that mean. All right. So typically how I would go about it. So as I said, summarize your results in a table format, and then describe that table shortly in paragraph form. So don't ever have a table or a scheme or a figure without really describing what's going on there. I mean, something doesn't serve a purpose unless you describe what it means. So if there's just a table there, then nobody knows what's going on in that table except you. So in other words, you can't really earn any marks in this context for it, but also in a real world context, nobody will ever then understand what you've done if you don't describe it at least in some way. You need to tabulate your observations. So like I've said a few times now, you tabulate your results. So tabulate the results, and more important, what is important is how you got those results. In other words, you say in the one column, what was the thing that you changed? What did that lead to? And then maybe something else if that is important or relevant chemical equation or whatever, link that with your paragraph. All right. You can also use the Postprac lecture to guide you on how to use your tables. That said, for experiments 1.4 and 1.5, it might be a bit difficult to use tables exactly, or might be redundant in the sense that there's very little information. You generally just use one or two equations, so there's not really information to tabulate or summarize in a table. But if you want to use a table, I'm not going to, it's always, that's dealer's choice. I mean, you can use whatever you would like to use to convey the information. So it's up to you. But I would use a table at least for experiments 1.1, 1.2, and 1.3 to summarize the information. Right. So now we'll talk about tables and how to properly include a table in your report and how to discuss it, introduce it, and how to use it. And maybe a tip on one or two of the tables that you might be using. So let's say you have a table and you've now nicely summarized your results. We've had a synthesis of 3 compounds, and we've included their structures, we've included their names, our product masses, the yields, and our purity of our final products, right? What we have. You always need to discuss that in some sort of chemical manner. In other words, we discuss it. I have some paragraph discussing my table, saying why is it important, what is the context, is it my own results, is it literature results, is it a combination of the two, is it a summary, is it just some data, is there data that has been processed into something else, what is it? And what is the significance of it, the chemical significance of it? No. That's what you need to be doing with your information. So if you're setting up your tables of your results for your manganese or your vanadium or all these experiments that you've been doing, try to find a nice way to summarize all the information in a table and then put that into context with your paragraphs. And don't be afraid to write a little and explain what's going on there. Because if someone just sees a table, they're usually overwhelmed and don't know what the table means. So that's where your words come in. You explain to them what your table means. Right. Just some little inconsistency. I see here I made a little inconsistency error. Don't do that. Don't be naughty. Okay, so that's tables and figures and schemes in essence. So the same, of course, applies to a table and a figure. If you have or figure or a scheme. If you have a figure or a scheme that summarizes something, it still explain what it is. Don't just put it there. And very importantly, don't just screenshot things from my lectures or the practical guide. In fact, that won't be marked if you just screenshot something from there. So please do your own work, make your own tables, make your own figures, make your own schemes, write your own reactions. You are now at a level where that should be something that you're able to do. So please spend some time and do that on your own. Don't just cheat. So speaking of tables, I'm going to give you some help and give you an example of one of the table that you may use. So you may use the basic idea for this one. So this is for experiment 1.2 for the manganese experiment. I would typically go about summarizing it as follows. So I would list the test tubes. I would say, what is the environment or the reaction conditions? How have we changed from the starting? So of course you have a paragraph here explaining, in this experiment, we do this and this and this and this. We change basic conditions, acidic conditions. We reduce it in basic or acidic conditions. You have a paragraph, right? Explaining how to start. Then you say, in table, we see that. Now you explain what in the table, right? So we're at the table. And now you look at it and I say, we have the test tubes, we have the environments, how we change the environments, and we have what we expect. In other words, from the scheme, what do we expect? Remember that big scheme that's in the practical guide as well as in the pre-prac lecture. And then we correlate that with the observed colors. So whatever you saw in the lab on the day, so colorless, orange, green. And then actually, so what does the observed manganese species, so how does these correlate? Colorless means +2, orange means +4, green means +6. So those two, for example, do not correlate. So in other words, in the discussion, that's something I'm going to discuss. I'm going to have to say whatever. I also make notes of what does each of these mean. So expected manganese species based on known redox reactions. Observed expected manganese species based on color observed in the test tube constituents. Yeah, so you can use this layout. Of course, it's a very basic layout, so you can elaborate or expand on this. So you can add some columns, some extra information if you would like. You can add pictures. I mean, that would be cool. If you actually took photos of the things that you did in the lab, you know, you can make it nice, right? So this is the basic layout, just to try and help you. And this is typically what you have, and you have a few paragraphs discussing what you saw in here, set the stage, explain what the table is going to do, give the table, discuss the table, right? So that's what I want you to do for experiments 1.1, 1.2, and 1.3. Specifically for 1.2, this is a very good starting point. And the other two, then you can sort of explain it in the same manner, or use a table. Maybe table for 1.3 won't be as intense as this one, but you can also use a table for that. Right. So I hope this has given you some chutzpah. I'm very motivated to go and do your work. Finally, the final section that needs to be in your report, or meaning the report writing aspect of your assessment, is then your reference section. So you get a few marks for references, and these references need to be correct according to the RSC referencing style. So of course, if you're using Mendeley, it's going to sort itself out. Otherwise, you need to look in the guide for how to do them consistently and ensure that all the references are correct. Okay, so that means you need to look at things like the spacing, full stops, italics, the bolding of numbers, etc. Right. And those are things that we covered in CMY 282. So I kind of see that as really covered. Not really something I need to emphasize again, but I mention it in the sense that you need to be aware that your references are going to count marks and they need to be consistent. All right. Finally, the second part of the assessment that counts the other 50 marks is your assignment. And my only tips here really are that you need to read these questions carefully. So the best thing to do is to have references for the assignment questions. So don't just try and do them on your own. You can look them up. You can look up what the answers are to these questions. So most of them you'll be able to find in some sort of textbook or on some academic journals. So look up the answers, and when you find the answer in one of those, just reference it. So in other words, rewrite it, reference it, and then you'll get the marks. You also need to use the mark allocation to guide you on how much you need to write. So if the question counts 16 marks, really don't just write 1 or 2 words or 1 or 2 sentences and a chemical equation expect to get the 16 marks. It means that there's, of course, a lot more depth than what you expect, right? So I hope that is enough information to give you sort of an idea on how to use the mark allocations. And then answering these questions is actually, so it's an assignment, right? So it's not just like a test question. So typically in a test, if something counts 2 marks, then you need to write 2 things to get those 2 marks. But now, because it's an assignment, it's more like a discussion. Each question is actually a discussion. So you need to properly explain what you are doing. It's not just answering the question. It's explaining where do we come from, where are we going, rationalizing your answer, why are you correct, why did you say that you said. So basically, in the same way that you would expect me to explain something to you, you need to answer these questions because you have the time. You have time at home to sit now and really type out. So I mentioned type out. So remember, everything needs to be typed. So your chemical equations need to be typed. And then if you have a reference, you need to reference it. So for example, if you're asked for a chemical equation, then you must say the balanced reaction equation for the oxidation of magnesium is as follows. So in other words, it shows that it needs to be balanced. It shows that this is an oxidation reaction, and then you give the reaction equation. So this is going to count 1 mark, even though you've given me 3 pieces of information in principle. So in a test, it might have just been asked to say, give the balanced reaction equation for the oxidation of magnesium, and then you get your mark for this thing because you have a limited time in a test to give an answer. But now you're at home, so you have time to really write me something or give me a well-formulated argument. In other words, you can write something proper. So that's what we expect of you to do in your reports. All right, so to answer these questions with great, you call it brevity. So in other words, really talk about it and talk and talk and talk. And there you can't really give enough information. That's actually the point. There's no such thing as too much information in these assignments. All right, so that's my final point. I wish you good luck, and I hope you've enjoyed your first experiment in CMY 285.